I'm going to talk today about an interesting part of the work of the UW that took place here and in Fort Detrick in Maryland, and involves the development of the botulinum toxin as a weapon of war, as an interesting research reagent, as a commercialized and very widely used and very with much income to the university. And then I'll tell you a story, if there's still time, about passion, love, murder that took place right here and had to do with botulinum toxin. So, botulinum toxin is the cause of a disease that was first recognized in the 1700s by Kerner. It was actually called Kerner's disease for a long time. Kerner was an interesting character who was both a poet, a published poet, and a doctor. And he treated a group of people that all had eaten spoiled sausage and all developed the same set of symptoms that included nausea, vomiting, weakness, and eventually, for some of them at least, real paralysis of their bodies. The agent he named the Wurstgift, the poison from Wurst, and it came to be recognized that there were three ways that the poison could get at you. One was when it was foodborne. There were several instances in the time between Kerner and now in which canned foods killed a lot of people by means of Clostridium botulinum. So there's the foodborne, there's the kind that develops in wounds. Even a scratch can actually be infected by the organism that causes this disease, and it can be fatal if not treated. And there's also a version of the disease that occurs in infants, usually pretty early in their lives. And again, if they are not recognized and treated quickly, their futures are doubtful. Here's a baby who has botulism, and you notice that the first thing you see is that the body is completely flexible, like a rag doll. And it's because all of the nerves that normally convey the signal to the muscles to contract, to stiffen, are non-functional. The botulinum toxin has caused them to be non-functional. You also see this in the face, and usually the facial effects are bilateral, which distinguishes them from what a stroke typically does, which normally will give you half of the face that is weak. There's a problem with vision, with eyelids. And the fatal part of this is that the failure to tell your diaphragm to move causes you not to be able to breathe, and you eventually suffocate and die of suffocation. There is some treatment, and it all depends on how heavily exposed you've been to the toxin, how the results will come out. But people who have the full-blown Kerner disease have to go someplace where there is a respirator and have the thing breathe for them for at least weeks and maybe months. The paralysis that is so striking and scary gets better over time, but that too takes a long time, sometimes months. And in the long run, people who have been treated properly and quickly eventually will recover from this disease. But if you're not treated, you'll die. The toxin is produced by a bacterium called Clostridium botulinum, which is a sporulating bacterium that prefers to grow without the presence of oxygen. And in the course of doing so, it forms a spore. This is the cell surface of the bacterium. This is the thick wall of the developing spore. And during the process of sporulation, the bacterium puts out this toxin into the growth medium, where it can be collected or where it can be obtained by a patient who unfortunately encounters it someplace. It's very easy in the laboratory to grow the organism, Clostridium botulinum, and to get it to make this toxin. And that's one of the scary things about botulinum toxin, is it is so very simple and so cheap to culture it. And when you do so, you have an incredible poison, as I'll show you shortly. So these are just two big jars this size. You give the organism a few things that it needs: yeast extract and sugar, and a few inorganic ions. You close it off so that there's no circulation of oxygen through it. These are anaerobes. And at the right time, you harvest it and collect from the medium this incredibly poisonous toxin. When I say incredibly, I really mean almost incredibly. Here are some well-known toxins: tetanus, diphtheria, ricin, sometimes used by the KGB to kill bad guys, cholera toxin, tetrodotoxin from the pufferfish, saxitoxin comes from Goniolax, which is a red tide organism that occasionally occurs on the East Coast and poisons fish with its toxin. We think of curare typically as being a very poisonous natural product, but it's 1,000,000 times less powerful than the botulinum toxin pound for pound. And sodium cyanide, which will kill you in 5 minutes, is 3,000,000 times less powerful than the toxin. So botulinum toxin is the champion, even more deadly than the tetanus toxin. So one thing that immediately occurs to anybody who looks at these facts is that these things could be weapons. And people have not overlooked that possibility in the past. They have been used, in several places over time, in small ways. They've never been really released as a poison during a war and during a big war, but they have been produced in large quantities by, among others, the United States of America. The weaponization of a toxin like this means getting it into a shape that it's stable enough, dissipated, can be easily disseminated. It's usually encountered by breathing it, so you want to have it in tiny pieces so that it can breathe in easily. So what you do with something like this is you freeze dry the agent. Now you have a chunk of something which you can powder by milling it into a fine powder, and now these things can be inhaled and will cause infection. There is also a problem that the toxin is a protein, and proteins are not in general infinitely stable. In fact, they're pretty unstable. So it is sometimes necessary with protein toxins to add something to the weaponized version that will help to stabilize the protein over time. And you want to do things that will allow you to put it on the storeroom shelf today, and it won't be clumped when you go back 6 months from now and need it. So weaponizing is a challenge, which was actually met or tackled by Baldwin, the Dean of the College of Agriculture here. You can't probably read this very well, but it describes how he was invited. He was at the time, he's a microbiologist who was at the time chairman of the microbiology department, or the bacteriology department. He was invited to a meeting of the National Academy of Sciences in Washington, sworn to secrecy, asked whether it was feasible to produce biological agents in large quantities, and when Baldwin said yes, he was the man. He went to Fort Detrick in 1942 and stayed there through the war, and supervised all of the biological warfare that went on in the United States. That included studies back then of anthrax, which has already appeared now in its ugly form a few years ago here. But this tells you something about the urgency of this. The likelihood was that if we didn't develop these things, somebody else would and use them. So even though morally it's not an easily defensible thing to do, in practical terms, an army to be ready has to be ready with this stuff. So they actually did something close to the Manhattan Project in biological warfare. They spent a lot of money, 10 million then was a lot of money. They scaled up the production of both anthrax and botulinum toxin so that they eventually had fermenters that would hold 250,000 gallons. And I'm telling you, that's a big fermenter, and could have produced pounds, maybe tons of the bad stuff, but it turned out that the war ended before this was accomplished.
May I ask a question?
Sure.
So this is going on for 18 months, that is 6 months after the war in Europe ended. So why did the Army, that takes you into the fall, and the end of the war with Japan?
Yes. So why didn't we stop instantly?
Yes, in August.
Well, there was no international agreement until 1974. I'll tell you about that in a bit.
Okay.
And I think it was the same situation where everybody said there were at least three countries that had developed this stuff: Germany, then Russia, and the United States. And everybody sort of agreed that if everybody else would throw theirs away, we'd throw ours away.
Yeah.
But it took an international agreement that came later during the Nixon administration to have that happen. I think also that these operations were huge, and so when one wound down, you couldn't just walk away from a huge fermenter. You have to do what it took to button it up and get it preserved. So I guess it's actually here that the dates are given. In 1969, the U.S. finally ratified the Geneva Protocol, which said no use of chemical or biological things in war. Nixon, during his years, signed an executive order and that said, we will no longer make this stuff, we will no longer store it, and all the stuff we have we'll throw away. That was idealistic, and it didn't actually turn out to be the case. When I came here in 1971, Ed Schatz, who's shown here as a young soldier, had just returned to Madison from having spent the war years in Fort Detrick, working with Baldwin on the production of war materials. Schatz came here in '69, I came in '71, and it was an open secret that there were stocks of serious toxins stashed away in several places on campus. One investigator had spent years and years isolating the saxitoxin that comes from red tide organism, and he just wasn't going to throw it away. And so it was supposed to be disposed of, and it may have eventually been, but not quickly. It goes almost without saying, but just to help you understand why such a big project got so much attention. The toxin is incredibly potent, as you've seen, and anybody who has a big bottle can actually make the stuff. All you need is the strain of that bacterium and a big bottle to grow it in, and you can make botulinum toxin. And in terms of effectiveness in a war, the people who were affected by this bug were taken out of action for a long time, so it really was an ideal kind of weapon. The weapon was actually used in Pancho Villa's years against federal troops. The story is that he buried pork and green beans for days, got the mixture to collect all kinds of foul stuff from the earth, and then used it to infect the bad guys, his opponents, with something that probably was botulinum. In World War II, by the end of the war, both U.S. and Russia had developed this stuff, and it was strongly suspected, but I don't know if it was ever established, that Germany had also done this. There are two interesting views of the use of biological toxins as weapons of war. One is a book, The Biology of Doom, which is clearly not, the author is no proponent of this. He's very much against this sort of thing. There is a different point of view given in a book, actually, that's developed from an oral history that Ira Baldwin participated in many years ago, but shortly after he came back from Fort Detrick. It's been edited and turned into essentially a biography. I've read it, and he clearly describes his belief that the U.S. really had to do this. There was no choice in the matter. So after the war, Ed Schatz, who had been in Fort Detrick for the whole period of the war, came back here and joined the bacteriology department. And he there had colleagues, Mike Foster, Hiroshi Sugiyama, who all were interested in the biology, and so the interesting biology of botulinum toxin. And these men, joined later by Eric Johnson, who's still with us here. I think Eric is retired, but he's still very much with us. They used the experience that they'd had at Fort Detrick in preparing this stuff to make enough of it so that they could target it as a research topic. And they did so very effectively, and so that it soon became clear how the thing did what it does. This is a picture that really strikes me. This is Ed Schatz in the foreground, Eric Johnson in the background. And Ed is holding a tube with his bare hand that contains 300 million doses of botulinum toxin. Those are therapeutic doses. As I'll show you later, it can be used therapeutically. But multiply the therapeutic amount by 10, and you're looking at the lethal amount. And so there's enough there to wipe out a good share of the western half of the country in this tube. Eric told me that during the period in the 70s, Schatz and his laboratory were essentially providing botulinum toxin to lots of labs around the country because they were so good at making it here. And he said that when they sent it, they just wrapped it up in a tight screw-top vial like this, put it in a cardboard roll, put a stamp on it, and sent it across the country. And that is almost inconceivable now. You would go to jail forever if you tried that at the moment. Having the luxury of a large university now to work on this botulinum toxin, it soon became possible to say that it was a protein of just a few subunits. These represent experiments in which a mixture of several proteins, a partially purified toxin, is placed on an electrophoretic apparatus, and the proteins are separated one from another according to their size from top to bottom. So this band is a protein, this is a protein, this is one. And what these represent are what they called disport. It was a form of the toxin that was not, they knew it was not completely clean. It has several major bands, one there, one there, there. So it was a very potent preparation, but not completely clean. This preparation, called Pure Tox, has only the two subunits that the purified protein has. And these names turn out to be important because they figured into the patenting of these agents. It turns out that if you're trying to patent a protein for use in humans, you can't describe it once and for all and saying this is the way we made it. And anybody who wants it can make it that way in the future. What you have to do is, to get FDA approval, is to make a huge batch of the stuff, and then that batch is the reagent, is the component that the FDA judges. And it turns out that each of these was a different preparation, differently treated by the FDA, and it turned out that there was some financial consequences for that. There are people on campus now who participated with people all over the world and tried to figure out exactly how the botulinum toxin worked. Tom Martin in biochem, Meyer Jackson in neurosciences, Cynthia Kachowski in neurosciences. All these people either use the neurotoxin in their research, or they are studying the mechanism by which it does what it does. This picture looks like too much, but it has all the players on this plate. We're talking about a nerve. This is nerve in the end of a nerve in brown, which reaches out and touches the surface of a muscle. And when the nerve has been stimulated, the muscle is told to contract. If you can't make that signal go, then the muscle will not contract. And the way that the contraction occurs is you start out with vesicles in the nerve that is going to touch the muscle. These are synaptic vesicles. They contain the neurotransmitter molecule. And under normal circumstances, that vesicle moves over, sits down next to the plasma membrane of this nerve cell, and with the help of a couple of very critical proteins, pulls the vesicle to the surface where it opens up and releases the neurotransmitter, which then diffuses across the small distance between the nerve and the muscle cell. These are muscle cells down here. That neurotransmitter goes across this narrow region, impinges on receptors on the muscle surface, and those receptors, when triggered, turn on contraction of the muscle. So these proteins grown in blue here, they're called snaps and snares for complex reasons and unimportant. Those are the targets of botulinum toxin. The toxin is an enzyme. And when it locks onto its target, which is these blue proteins, it cuts them with its activity as a protease, and they become useless. Without that, here's the example here. This shows the whole process by which the toxin enters the cell. It is taken in by endocytosis. Then it is released from the vesicle inside the cell and goes out and finds these snaps and snares, breaks them into little pieces, and now the secretory granule containing the acetylcholine can't fuse with the membrane, so the acetylcholine can't get out, so the muscles won't contract, and the symptom of the disease is this very, very relaxed muscle. Turns out that shortly after people quit using botulinum toxin to kill each other, they started using it to improve each other. And it turns out that there are a number of human diseases that are perfect as test places for the efficacy of these botulinum toxin things. Here, for example, is a woman who, before the treatment, had an eyelid that was drooped, and presumably because the muscle that normally holds it up was not being signaled from the nerve. And here they have injected just a very little bit of botulinum toxin into the region around that muscle. And it says September, I can't read how long it was, but typically it's a couple of weeks. Here's somebody whose forehead was terribly wrinkled and is completely straightened out after 2 weeks of treatment with Botox. It's turned out that there are many human diseases, some that you wouldn't predict, known what the mode of action of toxin is, that are either known to be susceptible to treatment with toxin or suspected to be. One of the suspects, for example, is migraine headaches. So people who have intractable migraine headaches and have tried everything are sometimes treated with a Botox injection in place that softens the contraction of the muscle in the back of the neck. And for some people who get no help from any of the medications, Botox is the answer. Not surprisingly, Botox is a hot item commercially, and it's an interesting story that, here's another case. BTX, of course, is Botox. People, as soon as they realized that this stuff was medically useful, began to try to fight over who found it and who developed it and who patented it and so on. The compound that was, the protein that was purified here, and as I showed you in a previous slide, was called oculinum. It was done so because Alan Scott, who was an ophthalmologist, I think in San Francisco, got some of the Botox from Ed Shantz here and used it to correct strabismus, the human disease in which the muscles and the position of the eyeball are too tight on one side. And an injection of this stuff every 6 months or so cures strabismus. So Alan Scott was the first one to show its human usefulness, but there quickly developed lots of people who were trying to get credit for having discovered this and get it patented. The big stumbling block was what I told you earlier, that the patenting of these things wasn't as easy as the patenting of a breakfast food. You had to have a huge batch of this, and the FDA had to approve that batch. And so there became various batches purified to various degrees in various laboratories, circulating around to be used for medicinal purposes, which was a trial basis. And it turned out that when Shantz and Johnson went to the wharf and asked about the possibility of patenting this stuff, Wharf turned it down. It turned out that there was a bad mistake. This is $1 billion business now, and Wharf declined partly because they didn't like the smell of the complication of purity, and partly because they already had one winner, which is called warfarin, and they didn't really want another poison on their shelf. And it wasn't at all clear when this first was approached whether there would be a useful application of the stuff. So Wharf didn't do it, and somebody else did. The people here found that they could stabilize the stuff they purified by adding another protein back to it. And Scott, who had been the physician who did the first treatments with this, had at least a part interest in the whole thing, and he sold his interest to a company, Allergan, and they renamed the compound Botox, and now Botox is on the market, and the UW product, Pure Tox, is, I think, on the market, although I actually was looking last night to see whether that's true yet or not. But there are lots of versions of Botox that have been patented and are being used around the world. So, I promised you sex and violence. And this is a story in which I actually had a role, so it'd be fun for me to tell it. There is a book called Winter of Frozen Dreams, which was written by Carl Harder, a Madison writer, a newspaper writer really, who documented the story that I'm going to tell you. The story for me began in 1978. I had arranged with this woman to come and work in my laboratory in the summer after the school year was out. And she was an honor student. She had a straight A in biochemistry. I think she was a junior in college when I met her. And so I expected her to come in the summer, and she never showed up. Eventually, I got a letter from her explaining that she'd gone a different direction, and that was the last I heard of her for a long time, until one day I opened up the State Journal and found her plastered across the front of the journal as the suspect in a murder. And it turned out that, you can't read that anyway. It turned out that what had happened was that she had become a prostitute to support a drug habit. And so she was, on the one hand, a straight A student, on the other hand, somebody addicted to drugs. She had found a boyfriend that was dumb enough to take out a life insurance policy of $1 million with her as a beneficiary after she assured him that he was her sweetie. There's no question about this. And it turned out that the cops found the boyfriend in a snowdrift out in Black Hawk, west of Madison, killed, they thought, with some kind of poison. This is a plot, but I don't think you need to see that. So Barbara Hoffman eventually was interviewed by the police. It looked very much as though she was the one who did it, but people couldn't understand how she managed to haul this big man down the stairs from State Street and out to the country. And then her boyfriend, her second boyfriend, showed up. Her real boyfriend, who had, it turns out, helped her haul the body down the stairs and out to the west of Madison. So now it looked like an open and shut case. She was going to be prosecuted for murder, no question about it, because they had a perfect witness who had been with her and had seen the whole thing. And then it turned out that the perfect witness also turned up dead in her bathtub. And so suddenly the best witness was missing. And to make a long, interesting story short, the police eventually figured out that these guys had both been killed with cyanide. And the thing devolved to a question of where did she find the cyanide? So the police came around to the biochemistry department and they asked each of us, do you have cyanide on your shelf? And everybody said, well, sure, everybody's got cyanide on their shelf. And they said, well, will you show it to me? And half of us went to where it should be and it wasn't there. That was not surprising because before this all happened, we were all pretty sloppy about such chemicals. Well, they assumed that she'd gotten the cyanide from one of us, and they weren't sure which one. And the trial began. I had to testify in the trial because I had presumably taught her how to kill with cyanide. I taught a course that she took, and in that course we talked about the biological effect of cyanide and the quick death that results from its consumption. But it was always a little unsatisfying to know that we couldn't pinpoint the source of the cyanide that she got. It turns out that in going through one of her check books, somebody looked at the stubs and found a check that was written to a chemical supply house in Milwaukee, and it turned out that she had bought a large jar of cyanide through the mail from a chemical supply house with no identification at all, just a check. So that really pinned it on her, and she was a goner. When she went to explain all this to the judge, she said that her real intention was not to use cyanide, but to use Clostridium botulinum. She had the idea that she'd take this boyfriend who had $1 million life insurance policy on her, take him for a honeymoon to Mexico, kill him off with Clostridium toxin, and have him buried there before anybody did any autopsies. And the only fly in the ointment here was that she couldn't get her hands on the strain of Clostridium that she needed to do this. She was smart enough to know that if she had the strain, she could easily make the poison. But it turns out that the people in the bacteriology department were more careful about their bacterial strains than we were about our cyanide, and she couldn't find it. The district attorney, when this case was tried, was good old Jim Doyle. The site of the action was an apartment above the State Street place, right one floor above the Sunprint Gallery. People at the gallery claimed that they heard a thump, 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 thump in the night. And here's old boyfriend. The boyfriend and the whole tawdry scheme was actually put into a movie. Now, I don't know if I can show you this or not. Let's see. Oops. It's a pretty bad movie.
What is it called? Winter Frozen Dreams?
I think it was that, yes. It's not going to let me do this. Let's see, maybe I can do it this way. I don't want you to miss this.
Are you listening? I'm leaving. I'm not going to work for you anymore.
What are you gonna do?
I want to be a good wife to you.
This is my fiance.
Boyfriend number one.
Are you really engaged to my
Number two.
Hi, I'm Barbara.
Linda Miller.
Miller. They are.
They wanted a $3 million policy.
Of course, I'm the sole beneficiary of that.
I love this. I'm fascinated by human beings. Teach me about the human mind. Tell me about guilt, rage, murder. Tell me about Barbara Hoffman.
I have a right to know. What's that?
You get the idea.
Shaved head, boxer shorts. Face looks like it's been sledgehammered.
Well, so she actually was tried, found guilty of both murders, sent off to Techita forever. And she has, since that day, this was in the 1970s, has never spoken to a reporter or anybody who was interested in this story. She's absolutely closed down. And she's still there. And that's my story of passion, sex, murder, and so on. Thank you. Questions about this?
Well,
Yeah.
what happened, if those of you in the biochemistry department were kind of sloppy about storing your cyanide, what was the theory, once it was discovered that she got her stuff from that lab, that place in Milwaukee, what were you folks saying? Where did
Well,
you-
the heat was off on us then, of course, but we all smartened up and started to lock up our cyanide.
All right.
But, you know, I shouldn't probably admit this, but laboratories are not safe. There's a lot of stuff there that if the wrong person got a hold of, it would be trouble. When I arrived in Madison in 1971, I took over a laboratory from a scientist who'd been here for years, Mark Johnson. And I found on his shelf a bottle this big, I mean, a really big bottle. And it was barbituric acid, a barbiturate. And it was used by a biochemist as a routine thing in the lab that was totally unrelated to its sedative properties. But there was enough stuff in that bottle to put out the city of Madison several times over, and it was sitting on a shelf. We're more careful now, have to be.
All right, I have another question. So the UW finally got around to patenting a version of Botox. So do you know if it's earning any money off?
I don't know that. I was looking last night to see if it was possible to find that. It's the brand that comes from here was not among the pictures I saw of,
Right,
of
right,
various
right.
kinds. But I don't know, and I'm sorry that I can't answer that.
Okay, here's my last question. So if, you mentioned a fellow who spent a lot of time tracking down the various places on campus where where chemicals had been stored that could have been used as biological weapons. Do any exist anywhere on campus now that you know of?
Yes, the really powerful toxins and poisons are now locked up. We've learned their lesson with that. But there are any number of chemicals on our storeroom shelves which, if ingested in quantity, would make you mighty sick or kill you. And you know that there are so many of them that we'd have to lock up half the chemicals in the place.
I know, but those, unlike the ones that I mentioned previously, have not been stabilized or prepared as biological weapons, correct?
That's right.
So, so what's the plan for these stashes of?
Oh, so for those stashes, you know, I've never had a stash like that myself, and I don't ask a lot of questions because it's a sensitive issue. I suspect that a lot of the stash disappeared when Shantz died. He was the one here who did this work.
Right.
So I imagine that in cleaning up his laboratory, whatever he had was properly disposed of. But there were pockets of this stuff all over the campus and around the country. I mean,
Sure.
This was precious experimental material. These people whose faces I showed you from here were using this as a reagent that was valuable. Now you can buy the stuff legitimately from a biochemical supply house, so there's some control over who has it and how much they have.
Okay.
All right, let's go enjoy the sun.